Hi, my name is Cameron. I'm a current junior here at Mass Maritime, and I'm majoring in marine science, safety, and environmental protection. And I'm also pursuing a minor in marine biology. And here in the aquaculture lab, I actually took on an independent study with sea scallop growth. And I started this project last summer, and I started with around 600 sea scallops, and I've been doing different sampling events to measure them and then put the data in the computer to compare over time. But thanks to the shellfish float, I was actually able to bring the sea scallops out in their natural environment, because in the lab they weren't doing very well and we were having a lot of mortalities. But again, once I got them out in the float, they're doing a lot better and they're thriving. And this is another opportunity that I've had to take on a hands-on experience, and a lot of the other students here as well have been working on different projects as well.
My name is Jack Garrier. I'm a junior student at Mass Maritime. I'm also in the MSSAP program. I'm also pursuing a minor in marine biology. I'm a Holling Scholar, so that's a student scholarship program that's provided through NOAA, and I'll be completing an internship through them this summer. One of the other species that will be growing in the Upwellers include Mercenaria mercenaria, or hard clams, quahogs, and out in that space we'll also be growing C. virginica, so that's oysters, and we're going to be using those to great effect, as part of research projects. And we'll be able to grow other species of bivalves in the future. And we'll be able to track dry weight and wet weight, and we'll be able to take mass measurements to be able to determine, how effective are the upwellers providing water flow and feed, throughout the season. So it's a very exciting project, and we're very proud to, have this opportunity.
So this is an example of a sampling event for sea scallop growth. We're taking scallops out of each basket and we're measuring them with length, width, and height. And then the other person is recording this data, and then we're later going to put that in the computer and then compare the data over time. And these scallops were some of them were initially grown in the lab, and some were actually off in the marine environment. So they're also separated by those groups in their baskets. So we're going to compare that data as well, and look from the data from last summer and compare it all the way to now. And we usually try to do a sampling event once a month at least.
So this is our water quality data song, and it's actively monitoring temperature, salinity, and chlorophyll content in the water column. And this is deployed in our shellfish upweller alongside the sea scallop experiment. So we're able to monitor those parameters directly live from our aquaculture lab because it's wireless. And through monitoring chlorophyll content, we're able to tell how much food is available to those shellfish, as the seasons progress. So we'll be able to track growth, and we'll be able to track all of these parameters in conjunction with the growth project. So it's a really valuable resource, and we're using it to full effect here for the shellfish upweller.
Hi, my name is Dr. Kristen Osborne. I teach in the Marine Science, Safety, and Environmental Protection Department. And this semester I'm teaching the Principles of Aquaculture class. And I am so excited about our new shellfish upwellers. We're able to grow all sorts of species. Here I have Chrysostrea virginica, or the eastern oyster, and we have about 30,000 of these juveniles that the class is going to be using, one, to understand the principles of farming them, and two, we're going to take it a step further with the science, and we're going to do a stocking density monitoring experiment. And so what that means is we're going to have so many of these little guys per unit volume, and we're going to compare how well they grow at really dense volumes versus more spaced out lower concentrations or lower density volumes. And so ultimately what that's going to tell us is how many shellfish can we put together in one bag that will still grow efficiently and would bring in a profit if we were to be farming them for profit. So it's just a simulation and educational exercise that students can become familiar with the principles of how you do this in the real world. And so with that, I would like to thank you so much again, Bilezikian Grant, for providing us these upwellers and the opportunity for the students to get real hands-on experience.
Well, one of the exciting parts about the entire Shellfish float upweller system is this simple basket. We actually have the students code them, so this is basket 5, 10, 15, 20, 25 is basket number 26. I have no idea why it's in the lab, but we'll get it back out there. This is the basket that instead of the expensive fiberglass custom bins that we're able to buy these commercially, and if this works out, and we think it'll work out pretty fine, future upweller systems will be able to be made in people's backyards, basically. So we have our custom designed upwellers. They're in place. They're all basketed out. You've heard Dr. Osborne's class is going to be using it. And then in the next few weeks, we're going to be putting it live online. A little bit of electronics, a little bit of magic, and the Wi-Fi, and we should have a signal coming out of it, and it'll give you the data from that data sound, and we're hoping to put an online live video camera off of it too. So again, thanks Bilezikian, you've given us a lot of projects. We made a lot of process in the projects, and hopefully we'll get you out here on the floats yourself. Thank you.
Thank you, Bilezikian Foundation.